it's really interesting, I think, that as technology advances, it gives us new opportunities to look at things in quite a different way.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I'm Kim Klinowski, your host for today's episode. Today's guest will explain how T cells are the ruckman of Aussie rules football, physical and ultimately controlling the flow of the response.
Our guest today is Dr. Stephen Turner, professor and head of the Department of Microbiology at the Biomedicine Discovery Institute of Monash University. Steve, thanks for being with us.
Well, thanks very much for the invitation. I love the description.
I tried. We may need an explanation of what a ruckman is.
Yeah, no, true. I actually use a different analogy for my lectures where it's like the Marvel sort of universe. So when I'm talking of adaptive immune system function, B cells tend to be Captain America because, the shield.
Oh, I like that.
Yeah. And then you've got Iron Man, who's the CD4 cell because they're sort of multifunctional and they think they're the boss. And then the CD8 cells, which I work on, they're the Hulk. They get angry and smash things.
Yeah, that's great.
But I do like the ruckman. I was the ruckman. I did play AFL footy, Aussie rules.
Were you the ruck?
Yeah, I was the ruck.
With your height, yeah.
Yeah, no, I used to smash things.
Okay.
Oh my gosh, I love it. Well, we will get to the science. Okay, but before we do so, the first thing that we like to ask all of our guests is to tell us a little bit about their pathway into science. So if you can tell us what got you interested in science from wherever you want to begin, kindergarten, high school,
Well, yeah,
college.
no, it's a really good question. And I'm actually asked that quite a bit because I think people see me and they think I should be an AFL footballer. How did I get into science? And it's actually growing up as a kid. I grew up in a very rural area of Australia. And I remember this one trip that I went with my family and we were going to a place in northern New South Wales from Victoria. So it was a long trip. But we went via Dubbo. And Dubbo is a place where CSIRO have a radio antenna for astrology and that sort of thing. And you might have seen it in The Dish.
Yeah.
Movie The Dish.
Yes.
Right. And I remember going to this place and we stopped off and visited, and like the CSIRO, which is basically the national sort of research organization, science organization, they had all these wonderful booklets and flyers, and I got absolutely caught up in it. And that's where I think my interest in science, and really it was a curiosity about how things work. I just wanted to know how things work. So, finished high school and look, I was half joking about the football thing, but I was actually on a bit of a career track to play football professionally. I got drafted in our system, but then I got down to sort of start training, and then I walked out onto the oval, and then I realized there was no way I was going to make it. Everyone was much better than I was. So I had to rethink what I was going to do when I grow up. So when I realized that wasn't going to be the thing for me, it was natural that science and going to university was what I wanted to do. I did a science degree at Monash, and really, again, I didn't really know what I was going to do when I grew up. So that's why doing a science degree, just getting a bit of a flavour of things. And I still remember the first time I walked into an immunology lecture, which was actually quite late in my undergraduate degree. It was third year. And I just fell in love with the immune system. I just thought, how cool is this system? Like, it's evolved so that we don't die. It is potent, tightly regulated, precise, coordinated, and I just loved that about it. And that's really how I got into science. And then, from there, I did a undergraduate research year, which is in our system called honours. And then if you do well enough in that, then that sort of sets you up to do graduate studies, which is then what I did. And it was just one of those things that I just kept following the science and, really loved doing it, still do. So that's sort of how I ended up where I am.
So somewhere along your path, you trained with a Nobel laureate, Peter Doherty.
Yes.
How did that come about, and what was it like?
Oh, well, it was amazing. How it came about, again, I don't, if people that know Peter, he's a very laconic sort of person. So there's no pretense, he doesn't sort of swan around. He's very down to earth. And I actually interviewed with Peter as soon as almost after I finished my grad studies. I actually visited the US. I interviewed at his lab at St. Jude in Memphis. But at that time, I decided not to come. My partner at the time, who's now my wife, she was still studying. So I decided to stay on for a little while longer. But it just so happened that Peter actually was returning to Australia for some time of the year at the institution where I was working. And so it was great because I got to catch up with him and we'd have chats. And then just one morning, getting a coffee in the tea room, we were chatting and he said, "Oh, look, I've got a position open. Would you consider coming over? It'd be great," because part of his plan was that after a little while he'd return to Australia and it'd be good to sort of have someone come back. So I said, yeah. So, it was completely fortuitous. I just happened to be at the right place at the right time. So I went to Memphis and worked with him for 2.5, 3 years there before we returned to Australia. And in terms of how I now approach my science and my mentorship, I took a lot from Peter. And there are all these sayings that I tell my students now that he used to tell us. So, for example, he'd always say there's no such thing as a bad result. There's only a bad experiment. And what he meant by that is like it really annoyed him when people were emotionally attached to an idea that wasn't supported by evidence. So, and I still remember this. I went in and sort of showed him some data that we generated and it didn't fit the hypothesis that we were sort of testing. And I was like, what have I done wrong? And he said, "You've done nothing wrong. That's the data." And he'd always tell me to commune with the data as if he was some sort of guru, which he kind of is, I guess. But what he meant by that is that actually, take in the data with no preconceived idea and let it tell you what you should do next. And that was a big learning lesson for me because, and this is what I try to instill in my students, is that even though we might be disappointed the experiment didn't work, it actually provides new opportunities, new ways of thinking. We have to kind of start to think outside the box. Well, what could be the explanation? And that's where new avenues of research open up. And that's the way it sort of works.
Either way, you learn something.
Absolutely. Yeah. I mean, it's just as important to learn what, doesn't work.
Yes.
As it does, how things work. And the other piece of advice, and he sort of said this half joking because, he's Peter, but he always said, surround yourself with people who make you look good or are smarter than you. And really what he meant by that is make sure that you have people that are invested in the same way that you are in the science, make the right sorts of connections that enable the work that you do. And really, as a mentor now, I see a large part of my role is sort of helping people realize that, making connections for them, helping them however I can, providing opportunity. Because in the end, if they do well, it makes me look good.
That's absolutely true. So you study T cells, which we haven't touched on as much as I would like to have on this podcast.
No wonder you invited me, Kim.
So can you just tell us what T cells do and maybe why they're so important for immune responses?
Yeah, well, T cells are a part of the adaptive immune system. And the particular cell type that I look at, the killer T cell or the CD8 cell, has a very important role in what we call immune surveillance. The reason it's important is because a lot of pathogens can get inside our cells and they're hidden. They're hidden from the immune system by being inside a cell. But our cells have a way of actually sampling what's inside them and then presenting that to the immune system. So, I don't know how much detail you need, but basically that presentation of what's inside a cell means that CD8 cells can kind of surveil the tissues and look for any hints of things that shouldn't be there. So if a virus gets in, for example, it'll make viral proteins that'll be chopped up into little bits and they'll be presented. So a T cell that is specific for that particular target can come along and surveil, have a look at what's on the surface, say, hang on, there's a virus in here. And then that triggers a program where they basically deliver this smash signal like the Hulk, where they smash the infected cell and it's removed. And the really cool thing is that this is very precise. Another analogy, which, it's a bit combative, but they're like a precision missile in a way, or a laser-guided missile. They can do this in a very precise way. So that precision means that you don't get extended damage to other cells that aren't infected. And then, other parts of the immune system can come up and sort of mop up. So that's sort of how I think about T cells, and particularly killer T cells.
So are there different kinds of killer T cells? Or they're only differentiated by their target or?
Yeah, again, so in terms of function, They all do the same thing. Right? But the adaptive immune system's evolved this beautiful, elegant system for generating specificity. So we have billions of cells circulating in our immune system. Each of those immune cells, T cells, have a unique receptor that is only specific for a particular pathogen. And they just basically lay in wait. So it's a preemptive kind of armament. When you get an infection, the cells that have the appropriate receptor can be selected out of that repertoire of cells that are in circulation and become activated. So they go from Bruce Banner to the Hulk.
I'm loving this analogy.
Well, it does, although, my son's a bit more of a kind of DC person, so he thinks I should be using Batman and Superman. But so in that way, there's this, very kind of exquisite specificity to it. So you can have the flu virus, for example, that'll get inside a cell. You'll have a set of cells that have the appropriate receptors that can detect flu, and they'll be the ones that'll help control that infection. You might get infected with, Epstein-Barr virus or, Ebola or something else like that. There'll be another set of cells out there that'll be specific for that, and they'll be recruited into the response to get rid of it.
So you may already have guessed that I'm not an immunologist, but I have another really basic question, and it relates to your mentioning that these T cells are surveilling. So let's use an example of a cell in the lung. How often is that cell surveilled? Are we talking once a day, twice a second?
In terms of that surveillance, it's getting the cells into the tissues, which is kind of important. Right. So if you've seen a virus for the first time, it actually takes some time to mount the sort of response. So you need to sort of pick out these cells that are responsible or going to be able to control the infection. You need to amplify them because they start off at small numbers,
right?
But then to sort of be able to clear an infection, you need much more substantial numbers. So they proliferate. So they clone themselves. And then what happens is that they can get into that tissue because of the infection. It actually triggers what we call an inflammatory response, which is sort of a first line defense. It's sort of like an alarm that goes off, and it goes off in the lung in the case of a lung infection. So cells that sort of are activated can find their way into that tissue by following the scent, basically, of this alarm or following the sound of the alarm. They get in there and then they'll start surveilling and killing off cells. The cool thing about adaptive immunity is the fact that once the virus is cleared, once the infection is controlled, you don't go back to the sort of state you started in. You get what we call immunological memory. And part of that is basically the placement of cells that stay in the tissues as sort of sentinels or guards. And we call that resident memory because they're in the tissues and they stay there. And these guys do sort of surveil. Now, how often it happens, I don't have the exact numbers, but often enough that if you get reinfected, they're right there ready to go, and they'll try and sort of knock the infection on the head. And that protective response is actually why we don't get infected all the time. So we're invaded with pathogens all the time. I mean, you take a breath and you're probably pulling in dozens of viruses. If some of them try to infect you, in the main, you've got some pre-existing immunity there that'll basically stop things before they get out of control. Sometimes we do get infected by viruses that can replicate really quick. It can overwhelm that sort of first line of defense or that sort of initial surveillance. But then it buys enough time for the immune response to get reactivated again. And you generate what we call a secondary response, and then they can come in and sort of mop things up. So you might get sick, but you won't be as sick as the first time.
Right.
And that's why vaccination is so important. What we're really trying to do with vaccination is set up that immunological memory response, that sort of protective state. We've tricked the body into thinking we've been infected already. And then that means that when you actually do get infected, you've got this kind of armament, this sort of defense that's ready to go.
Yeah, so I think we've gotten a good background on T cells so far. So why don't you tell us about the main question that you're asking regarding T cell immunity?
Yeah, so the question that I'm asking is really, how do these cells go from this what we call naive state where they don't have any function? How do they acquire that function? And then how do they maintain it in this sort of memory state? So one of the characteristics of immunological memory is this rapid responsiveness. So they can mediate this protective response very quickly without the need to kind of acquire that function through differentiation again. And it's just about. So what my lab looks at is the programming, the genetic programming that underpins all that. We look at how the genome within the nucleus of a cell is organized and remodeled and positioned that then not only enables this transition of cells from this sort of ineffective to an effective state, but then how that is then stably maintained in the long term. It's interesting because, understanding what we call the epigenome, that is how the structures within the nucleus are sort of organized in a way that enable kind of a cell to do what it is really important. So the genetic information in a cell, in our cells, is the same genetic code is there. So if we look at T cells, they have the same genes as a muscle cell. But the genes that are utilized to give you a muscle cell versus a T cell are quite distinct. And that's because the way that the genome is organized is in a way that's such that you only get muscle cell genes turned on in a muscle cell and vice versa in a T cell. So what my lab really looks at is that organization. How is it that a T cell can reorganize its genome in a way that then turns on the genes needed to combat infection?
And this reorganization, what's involved with that?
Yeah, so really the way that a cell turns on and turns off genes is at a very basic level is through what we call remodeling. So the nucleus of a cell is very small. It's a very small space, and there's over 3 billion base pairs of DNA in there. If you stretch out DNA, it's about 2 meters long. So, higher sort of organisms, higher order organisms, metazoans, have worked out a way of compacting that material in a very sort of tightly compact way, so you can fit all that genetic material in a very small space in the nucleus. So that leads to sort of this structure, which is basically really compacted and tightly put together. But that is not very conducive for turning genes on. And the reason for that is that to turn a gene on, you actually need to open up those structures so that the DNA that you need to be transcribed can be accessed by factors that can drive the transcription of that gene. So basically that opening and closing of the genome is really at a base level how genes are turned on and off. And that mechanism by which, a cell, or a T cell, turns on the genes needed to become a Hulk cell, a killer cell, is through the opening up of genes that needs to turn on. So what my lab looks at is, well, what are the sort of mechanisms that you can get these changes in structure from a closed structure, which is transcriptionally silent, where genes aren't being turned on, to one that's more open, where those genes are going to be turned on. And really the question is twofold. One is, how does a cell know which genes to target? All right, so that's usually through the expression of particular factors we call transcription factors that actually bind DNA sequence. So within, say, for example, a gene called the interferon gamma gene, which is one of those factors produced by these killer T cells to combat infection, there's a particular DNA sequence that can be recognized by a particular transcription factor. So we study basically how then that part of the genome is opened up that allows the factor to bind. So that's how a T cell knows what to turn on. It has factors that can recognize the DNA, and when it opens up, it'll turn a gene on.
So are the different genes that need to be turned on, are they physically close on the same chromosome, or are they on different?
No, they're all over the place. So that's really one of the interesting things, right? So, I can't remember how many chromosomes we've got. What is it, 24? I have to go back.
It's so funny how when over time you get so down in the weeds in your own research, you're like trying to recall basic biology and you're like, wait a minute.
Someone might call me out. There are several. Yeah, exactly. So a really interesting question is, the genes that a T cell needs to mediate its function, to do its job, they're actually found all over the place. There are multiple locations within the genome. So that is one interesting question. How is it that an immune cell like a T cell can coordinate turning on all these genes in different places at the same time? And that's one thing that is of particular interest to us.
Right. And it's mostly, I assume, the T cell getting signals or acknowledging changes in its environment that kind of directs this. And I imagine that maybe it's those signals that are very different for what's programming through these different.
Yeah, that's exactly right, Kim. So, the way that these things that conducted, I guess.
Mhm.
You can sort of imagine like there's an orchestra, which is sort of the genome. You need different parts of the orchestra to sort of play together. You need a conductor, and that conductor is actually the signals that that cell will receive. So there's multiple signals. So there's the recognition event. So that antigen receptor is one of the signals. So that tells you that there's an infection. The inflammation that I talked about, so the ability to recruit a cell is also another signal that says, yep, this is where the specific cell is you need to target. There's an infection going on,
Mhm.
right? So we need another part of the orchestra to kick in. So, one of the areas of research that we're really interested in now is how do all these things converge, and how are they integrated if you've got multiple sort of signals that are directing things? How does a T cell kind of integrate all that information to then mediate the function that you need?
So one thing that strikes me is that your description of the changes when we go from a naive T cell, there's genomic architecture changes, and we end up with an effector cell or a memory cell. That's kind of reminiscent to me of something else I don't know a lot about, but stem cells.
Oh, okay.
Where stem cells differentiate and they undergo, changes in their gene program expression. And is that a valid?
No, it's absolutely, it's a great analogy. And really what we are talking about here is sort of a fundamental of cell biology for that reason. We're talking about cell fate decisions, where a cell will have potential, and then depending on what that potential will be, whether it'll be an AFL footballer or a scientist, will really depend on the experience, and what sort of that cell receives. So it's absolutely analogous to that. And in fact, a lot of the research questions that we're now addressing, we take our sort of cues from other research areas such as stem cell biology. And the really cool thing about it actually is that you can go right back. So a lot of the mechanisms that we're studying are really evolutionarily conserved. So the sorts of things that yeast do to regulate their gene function through this sort of remodeling of genome is exactly the sort of thing that mammals use, or humans use.
If it works, use it.
Yeah, no, that's right.
So I think it's maybe it's not, so we should point this out. So understanding what these signals are and how they're going to program the T cell is going to be important for directing T cells to certain functions depending
Yes.
on what you need, right? So
Exactly.
is this kind of what we're thinking about? I know there was a big push for a universal flu vaccine, with maybe T cells being the star of that show. Is the idea of using that kind of information to generate a universal vaccine maybe?
Yeah, maybe. Although I think, in that case, it's sort of, can you kind of select a target that's shared between lots of different strains of flu or subtypes of flu and get a T cell response? And the reason the T cells are important is the targets that they recognize are much more conserved. So it's less about the function per se. It's more about being able to kind of prime a response where that T cell is going to be able to recognize
Multiple
targets because they're all shared. But I think where it is really important, this sort of programming idea, is in instances where we're trying to develop new immunotherapies for things like cancer.
Right.
So a good example, of course, is checkpoint blockade. So what checkpoint blockade is it's a therapy where in many people, the T cell response to the cancer, which, so T cells are also important because cancers are presenting basically things that shouldn't be there because they're cancerous. CD8 T cells can actually recognize that and often control cancers. But when that gets out of control, of course, that's when it's a problem. And one of the ways it gets out of control is that the cancer can actually turn off the T cell response. And they do that through the expression of what we call checkpoint inhibitors. So there's receptors on the immune cell, the CD8 cell, which actually are a natural part of their function anyway, to turn them off. And the reason it's a part of their normal function is that T cells, like I said, are very potent. You don't want them out of control. And one way the immune system basically regulates that is through the upregulation of these checkpoint inhibitors to turn the immune response off at the right time. So you don't get an immune response that gets out of control and where you don't need it.
So it's like waving at the Hulk and saying, I'm a good guy.
Exactly. Or it's, just calming him down, so he turns back into Bruce Banner. That's exactly right. So, in a lot of instances, in a lot of cancers, that they actually have selected basically this immune escape mechanism where they can kind of actively engage those checkpoint inhibitors, and they inhibit the function of the cells. So checkpoint therapy is basically the administration of antibodies that interfere with that repression, and you reactivate the cells, you reanimate them. Now, the problem is that while it works in some cases, it doesn't work in others. It's actually there's quite significant side effects. It only works for certain types of cancer. But the real downside is that you can reactivate those cells, but they become dysfunctional again. So you can't maintain the function for very long. So a big push is, can we actually, by understanding the sorts of basic mechanisms that we're studying in the lab, can we apply that to reprogram cells so that we make them memory cells? If we understand those mechanisms, what's turned on and off, can we reshape it? Things like using CAR therapies, which is another immunotherapy where you can sort of reprogram cells or redirect them by putting in different sorts of receptors that actually target particular tumors. Again, they've proved particularly effective, particularly for blood cancers. But again, there's a downside is that they don't persist. So they come in, they do their job, but you'll get some kind of regression of the tumor often. It can be quite successful, but you can get recurrence because basically the cells can't be maintained. So again, by understanding these kind of mechanisms, can we reprogram these cells down a path where they can be maintained, do their job, and then protect the person through this surveillance mechanism after the fact.
So you describe using a lot of different techniques, quite an array of techniques. You're looking at genomics, systems biology, you've got recombinant viruses, you've got special mouse cell lines. That's a diverse set of expertise. How do you get all that expertise within your research staff?
Yeah, I don't. There's one answer to that. One of the things about working at where I am at Monash is the institute that I'm within is it's really one-stop shop. So if I need a particular expertise or technology, I can walk down the hallway and there'll be someone that'll know what they're doing. So we collaborate. Yes, and this is one of the benefits of, one of the joys of science is being able to meet people who are smarter than you, make you look good, and help, that it's enabling. The other thing, though, I think, is that I have sort of been quite strategic in determining what I think are going to be the workhorse technologies. It's really interesting, I think, that as technology advances, it gives us new opportunities to look at things in quite a different way and learn things that we might have assumed we know, but then when you sort of use a different way of approaching it, you get a completely different answer. And again, this is one thing that I think, I learned from Peter. So when I came to his lab, we had no tools to identify these virus-specific cells directly out of an animal. So we had to do these kind of what we call limiting dilution assays, where there was like plates and plates of tissue culture, where you'd basically sort of dilute the cells out so there was one cell per watt, like it was a nightmare. And then these reagents came along called tetramers. And really what they are, they're just little protein structures that are the targets that the T cell sees, but they've been basically now put into a form where we can basically throw them on the population of cells that we're looking at. They'll bind, and then because they're fluorescently labeled, we can then run them on the machine and detect them. Completely revolutionized the field. Because for the first time, we could directly count how many cells there were.
And specific cells.
And specific cells, yeah, that's right. So specific cells. And, we'd made these sort of estimates using this old technique, and we were orders of magnitude out. All right, because, there was limitations to the tissue culture strategy. But the other advantage it gave us is now, because we could identify these specific cells responding to a particular infection, we could isolate them. We could use, flow cytometric techniques to pull them out, and then we can interrogate them. So then we can apply all our sort of molecular biology or functional assays and really drill down into what they're doing and what makes them up. And that's really what we've done. The other thing I would say is that, being I'm a bit of an early adopter of technology. I like to sort of get in early. So we were doing this technique called single-cell RNA sequencing. And in fact, I was sort of doing multiplex PCR, which is sort of a polymerase chain reaction amplification of genes. Was a very old way of actually looking at what genes are turned on and off, where you basically can isolate the RNA, you do this sort of reaction, and then you can measure it on a gel. We used to do it one gene at a time, and then I adapted it to be able to do multiple genes at a time. And then I adapted the tetramers, because we had those, we were able to sort of sort individual cells out. So we could look at this sort of multiple genes simultaneously from a single cell. And then this technique called single-cell RNA sequencing came along, where now you can do the whole transcriptome. So the whole sort of atlas of genes that's within a cell being transcribed, you can now detect. It's, mind-blowing. And it was mind-blowing, 5 years ago, but now it's a standard technique.
Yeah. It's
Right? It's
so fast.
So I think that's the other thing. We use a lot of what look like technologically advanced techniques, and they are, but they're so quickly being adopted that they're kind of now standard tools that we would teach students as they come in.
Yeah, I remember the first time I saw a graph, like one of the UMAP graphs of a single cell RNA-seq experiment. It's like, okay, what the heck is that? But now you see it frequently.
I still think the question is what the heck is that is still a valid question, because
Yeah.
I don't think even people understand somebody. But I mean, a really good example is sequencing, right? When I was a grad student, I would pour my own sequencing gels, knocking out the air bubbles.
Yes.
You'd have the single lanes for each kind of nucleotide. And students now.
Clamp your gel or it would leak.
Yeah. No, that's right. Gaffer tape, the whole thing.
When you pull the glass plates apart and it sticks to both, rims down.
So you know what I'm talking. So, but that was the way we did things,
Mm-hmm.
right? But then now, most people, you make a library and you shoot it off to the facility.
Right.
I mean, so things, evolve, they advance. So it sounds like so even sort of genome editing, that's a thing, like it's a standard thing now. So a lot of the technologies we use, we're interested in knocking out genes. If we've identified something that we're interested in, one way to test its function is to remove it from the T cell and see what the impact is on the immune response. It used to be you'd have to make knockout mice, which was a whole thing. But now you can take the cells, you use a sort of, what's called CRISPR technology, where you can target deletion in a very specific way. And that's a standard technique now. We do it all the time. It's a lot cheaper than making mice.
And after you knock it out, you can do single cell RNA. So you can
And all those things.
That's
All those things.
Exactly.
So I have a question. Maybe it's been on my mind lately. But what is the funding structure in Australia? How are you funded? Are you concerned about any funding challenges that may be ahead that may mirror what is currently happening in the United States of America?
Yeah, look, I think, touch wood, it's not nearly as severe as it's critically seems to have got recently in the US. So my lab is funded primarily through research grants, so government-funded agencies. We have the National Health and Medical Research Council and the Australian Research Council, which are our main bodies, and then a bit of industry and philanthropic sort of stuff. But it's nothing at the scale that you see here in the US. And funding is hard. The success rate in Australia for these sort of standard grants is about in the order of anywhere between 12 to 15% success rate. There are some other avenues. So there's, consortia type grants, but they're very difficult to get.
Mm-hmm.
And it's an issue. It really is an issue. I think, if you sort of look at the data, there's been an attrition of funding in Australia. So it's not like there's been cuts per se, but the budget for research funding hasn't grown, even commensurate with inflation.
Mm-hmm.
So if you sort of take it in real terms, like dollar terms, value for money, we're sort of down maybe 15, 20% of where we should be, accounting for that. And science is expensive, especially in Australia. Right? I mean, it's probably going to get more expensive now with tariffs and whatnot.
Sorry about that.
Oh, well, what are you going to do? So, we have limited resources and you have to be quite resourceful. But I think, one of the things it does do is focus your thinking.
Mm-hmm.
You really kind of think about the sorts of experiments you're going to do and how you're going to do them. The collaborative nature that I sort of touched on before is also very important because you can pool resources, pool intellect, particularly with postdocs and students, So it's sort of forcing people in a way, but it's sort of the thing that we were doing anyway. But I think it's an issue. I think, and it's interesting because, we've got a federal election coming up this year. I've spoken to sort of our equivalent of Congress people, senators. So they're our sort of local representatives. And science research and funding for research isn't even on their radar. They appreciate how important it is, but they, the way it was described to me is they don't get letters about research funding. They get letters about, cost of living and the price of houses and immigration. So I think a real challenge, and something that we're actually starting to do a lot more of, is, research scientists need to really be strong advocates, not just for the sector, but also communicating just how important basic research, medical research in particular in my case, is for the functioning of a society, of a community. And it's interesting, anecdotally, if you talk to someone off the street and they ask you what you do, you sort of explain it, they get it. I think in the main they get it, they appreciate it, and they value it.
Mm-hmm.
So it's being able to somehow convince the politicians that this is something worth investing in a much more significant way than it currently is.
Mm-hmm.
So we're in this sort of, I won't say it's status quo, because there is this kind of attrition that's happening. And it's impacting. I mean, we can sort of see it. There's a lot more sort of job insecurity, particularly for ECRs and mid-career researchers. We have, we're not a very big country by population standards. There's not a lot of tenured positions at universities, for example. So we're seeing a bit of loss in the sector, a lot of kind of brain drain, we call it. There used to be, people would be kind of coming to the US. Might be reversed now.
Exactly. I was maybe asking for a reason.
Do you have any openings at Monash?
Oh, yeah, we're always looking.
Okay, so moving on from science, and when you're not thinking about T cells or influenza, what are you doing?
Yeah. Well, I love golf. One of the reasons I've swung past UGA is not just to see some great friends and colleagues, but also to play some golf. So my kids are now 16 and 14. They're at an age where they've got some interests that overlap. So they're right into music, particularly metal. So my daughter plays guitar. My son plays bass and drums. So when I'm not working, I go to music festivals with them. We jam. So I play guitar as well.
Oh, that's cool.
Yeah, no, my daughter won't let me play lead though. She insists I'm just rhythm. Yes, no, and I love to cook too. I love to bake. I love a tart.
Ah.
Yeah, I love a cake. So during COVID particularly, I went through sort of the Nigella Lawson sort of recipes. And one of, if I ever have one claim to fame, it's not the science papers or anything that I publish, or students don't tell them this, but even the students graduating, but I got a shout out from Nigella Lawson because I did one of her toffee puddings and I posted it and she replied.
I love that. I love that.
Now, during the pandemic, did you also have your own sourdough starter? Because that seemed to be required for everyone.
No, we didn't do the sourdough thing. We did a lot of bread baking though, but I'm more of a dessert person. So there was a lot of lemon tarts, a lot of marshmallow things.
Sounds wonderful.
Any pavlovas?
Lots of pavlovas. Actually, my wife, Nicole, she's the pavlova queen. She's had a recipe that's been kind of inherited down the generations. So she won't let me do pav. She's the
we may have to get that recipe. So our guest today is Dr. Stephen Turner, professor and head of the Department of Microbiology at the Biomedicine Discovery Institute of Monash University. Steve, thanks for being with us.
Thank you.
Links to additional information about Dr. Turner's research can be found on our Instagram account at PPPPodcastUGA. As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched this season. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Sid Wiggin, for research, editing, scheduling, and keeping us on track.